with Richard Mendel, MD, a neurosurgeon from St. Petersburg, Florida. We're also joined by two distinguished panel members that may expand, and we'll introduce them, and then we'll turn it over to Richard. Good day, Manuel.
Hello, nice to meet you. Pleasure being here.
Manuel's a neurologist from Oviedo, Spain. He put together a dynamite hangout conference last month. And Simon Down. Simon, good day.
Hello, nice to be here. It's 4 a.m. in sunny Tokyo.
Oh. Well, Richard, he's leading the hours of a neurosurgeon.
Yeah.
I'm practicing.
And,
wonderful to be here. Thank you.
What's that, Simon?
Great to be here. Thank you.
You're welcome, Simon. Okay, Richard, it's all yours.
Okay. You know, I was just going to try to wrap this up with just the third talk, and it just kind of touches on a few of the issues at play now. Okay, so at the end of last week, we were talking about the tissue-specific stem cells, and we talked about how, you know, biology is really going to become quantitative now, and much more mathematical. Just trying to measure things like cell turnover and embryogenesis and all the things that are remarkably quantitatively linked, but only now do we have models of it, just like protein folding patterns and the way the brain itself folds. So all those are going to become numbers intensive in the near future. And then we also talked about, 1 second, let's see. I'm not able to advance this right now. I don't know why.
Click on the slide itself, I think. It worked before.
Hmm.
Go to the column on the left and click on the slide itself. I think that worked.
Yeah, I've been doing that.
Oh, okay.
That's what's not working.
Oh, okay. Yeah, we'll get it. Yeah, maybe try to go back. And we'll take this commercial break to talk about Vlad.
There we go. Wait.
Oh, they got it?
Maybe. Let me try it again.
Okay, yeah, just start from the beginning.
Yeah, so what we were talking about last time was, you know, the quantitative aspects. And so we also talked about, you know, the quote-unquote potential therapies. And let's see. You know, we talked about different targets, and the targets were kind of difficult. You know, we mentioned diabetes, Parkinson's disease, heart disease, spinal repair, which I told you I thought was pretty surprising because that seems just so complex right now, and also retinal degeneration. So the potential therapies, in trying to predict them, you know, you think about it, we're really not very good at predicting what treatments or what diseases we're going to be able to fix. You know, because I told you when we were talking about all the genetics in the 90s, we thought cystic fibrosis, you know, a one deletion problem would be easily fixed. And really, we haven't made any headway with that because we didn't know about all the epigenetics that went with it. So the thing that really changes quickly is technology. So we're not really sure what technologies are going to change everything, but the changes come quickly. And you know, for instance, just think of that Keystone Pipeline. The technologies in fracking were unheard of at the beginning of the pipeline construction, And here we are now. We don't even need that extra fuel because we have so much of an abundance with the fracking. So, the quantitative biology and the cell turnover studies, they're going to be a big deal. Now, one of the most successful parts of gene therapy has been this hematopoietic stem cell success. So this FACS, the fluorescence activated cell sorting, it's a machine, and it was developed and really fine-tuned by this fellow Irvin Wiseman at Stanford. And what happens is the machine sorts a complex mixture of heme into a single cell based on just cell surface molecules. And so we're much, much better now at rescuing people that have like myeloid tumors or, lymphomas or myelogenous cancers. Because if you remember 20 years ago, they used to have the leucovorin rescue. Remember that people would be radiated and their entire bone marrow destroyed, and they would get this leucovorin rescue, and if they were almost dead, and if they responded to the leucovorin, it was like Lazarus, But now, with a lot of these myelogenous cancers, we're so good at preparing the cells to replace, because once you instill those myeloid cells, everything sets back up again. The T cells, the B cells, the NK cells, all the immunosuppression system, recreates itself. And it does so very specifically based on the fact that you could really select out a single cell at a time with this fluorescence testing. Now, this is really very interesting, and I just learned this week. I was looking at cell turnovers, and it turns out that, unfortunately, starting in the 40s and up until this test ban treaty in 1963, the U.S., USSR, Britain, and France admittedly were conducting experiments where they were doing radiolabeling when they were detonating nuclear bombs and atomic bombs, and they had no consent whatsoever to do that. And what had happened was two researchers at the Karolinska Institute figured out a novel way to measure the ratio of carbon-14 in the embryonic cells, the cells that weren't felt to reproduce or proliferate after birth. And so they could measure the quantity of C14 in the cerebellum, in the cortex, in the bones of patients, that were exposed to all this radiation all over the planet. And so using this radiolabeling technique, which is apparently really very sophisticated, there's an idea of where the slow turnover of cells in humans are. I mean, this is a live study basically conducted in humans, and the people at the Karolinska Institute in Sweden figured out, a mathematical model to measure the ratio of carbon-14 to carbon-12, and therefore know what cell populations were proliferating and which ones weren't. So it turns out that in the brain, the only stem cells are in the layers surrounding the lateral ventricles and in the dentate gyrus right next to the hippocampus. And it also seems that there are these things called neurospheres. And these are stem cells that are able to be grown in a tissue culture. So remember, it's in vitro, it's not in vivo. So these stem cells are harvested from the brain in the paraventricular space of the lateral ventricles, or in the dentate gyrus at in the hippocampus. And these cells can be subjected to or goaded into proliferating. And the interesting thing about these is that these cells actually do self-renew. And so the idea is that even though many other cells don't self-renew, for some reason there are neuronal cells that do renew. And so this is apparently going to be, is really being looked at as a source of expandable cell populations for treating neurodegenerative diseases. So another issue was, according to one of the people that at the University of Minnesota, there was this impression that because of the way the science of developmental biology is, that it's really unlikely that there are these like unicorn cells, these embryonic stem cells that we've come to think of as the archetypical cell that can evolve into any cell line. The idea is that it's pretty unusual. It would be pretty unusual to think that there are embryonic stem cell type of stem cells in a full-term human. It would be more likely that these in embryogenesis there would already have been these cells would already be programmed to at least develop somewhat along a different pathway into something specific, like a mesenchymal layer, etc. But it seems there are these multipotent, but not pluripotent cells that can get tricked into proliferating in culture. So what that means is that remember, we're trying to use that phrase, induced pluripotent cell, rather than calling it an adult stem cell, because there's probably no such thing as an adult stem cell. There is probably cells that can be directed down a pathway that maybe it wasn't going to follow it by manipulating it one way or the other. But these are all, remember, in vitro, not in vivo. So when you see these brochures of people come into your office and tell you about amniotic fluid or stem cells here or there, just remember that it's very unlikely that these things are going to be efficacious. This is more of what was phrased as aspirational stem cell. What do they call it? Stem cell shopping or something like that. But the idea and what happens when companies are trying to push something, they'll use the example of mesenchymal stem cells, which in cultures and with the right growth factors can be manipulated into a proliferative or an amplifying cell for a particular line of cells. And another phrase from mesenchymal stem cells that's used is stromal stem cells. Now, one of the things that we really didn't talk about is the in vitro fertilization clinics. Because when you think about it, we've talked quite a bit about stem cells and these pre-implantation embryos, and what John mentioned, which is the notion of a conceptus versus a embryo and a portion of the placenta. Where is all that stuff coming from? And where it comes from is in vitro fertilization clinics. And so it seems like these fertilization clinics, the in vitro clinics, have really become a giant industry over the rich and middle income worlds. And what happens is that all of these IVF clinics have large numbers of embryos that are, you know, frozen in liquid nitrogen. And if the patient that donated the sperm, the egg, you know, they have a successful course and they have a child after 1 or 2 trials or 2 or 3 trials, there may be, you know, dozens of embryos left that are frozen in liquid nitrogen. So the question becomes, you know, what do you do with them? You know, do they get thrown out or destroyed, or do they get donated to research to establish other cells, you know, stem cell lines? And so I was kind of looking at some of the laws, and it's funny. In the USA, there are no federal prohibitions of any type of stem cell research, but there are restrictions on the way in which public funds can be spent on research, especially in the way in which the NIH grants are distributed. So I think that, you know, where stem cells are coming from, the source of where they're coming from, is really very interesting. You know, because, you know, we're not really. I wasn't clear that they were all coming from in vitro fertilization clinics, but it sounds like the vast majority of them are. And the fact that there's no federal prohibitions on stem cell research also came as a surprise to me, and it kind of reminded me of the HIPAA laws and Stark laws. Really, more the Stark laws. Like, nobody really understands what Stark laws are, and I took a course at Auburn, a really interesting course, and it turns out, you know, Stark laws it's just a payment rule. No one ever goes to jail for Stark laws. Now, doing something that constitutes Medicare and Medicaid fraud, that'll get you in jail. But a Stark law violation is not a big deal at all, but it's very misunderstood. And I think that probably most of us don't really understand the laws governing stem cell research. And like I said in Talk 1, I wasn't getting into any of the research, ethics, or religious ends of things. But I did think it was interesting that so much of this, you know, the resources come from IVF clinics, and that's something I was completely unaware of. But all in all, I think that about wraps things up. I would think now that, you know, recalling the difference between what people portray as clinical results is easily separated once you start thinking about, A, was it really in vivo or was it in vitro? And if it's in vitro, the odds to being able to translate it in vivo are probably pretty small in a typical clinic setting that we see today. And let's see. So the other thing was to keep in mind that you want to know when experiments are done on germline cells, sex cells, or whether they're just done on the somatic portion of the genome. And I think that makes gives you a much better structure to work with to understand what people are trying to explain when they're talking about clinical rationale for these stem cells. But I really think that's pretty much all I had for today, unless somebody has any questions about it.
Yeah, thank you very much, Richard, for a very clear presentation. Also, let me start things off. Just to be clear, Richard, what are the areas of contention in stem cell research that you hear about religious groups getting all upset? What are the areas they object to? Are they the intravitreal, the clinics, or the existence of the clinics, or just the fact that any research is being done at all?
Well, you know, I think, unfortunately, and often with religious groups, they hear terms getting thrown around that are very, worrisome to them, and I'm not sure everybody understands where we are with the stem cells at this point in time. You know, you're taking fertilized embryos that are not wanted by the parents because they've already maybe conceived a child, and these embryos are being donated for research. Now, these are not, well, they are, I guess technically they would be viable put in the right environment.
Right.
But remember, we talked about nothing is ever done on a embryo that has reached that primitive streak phase, you know, where
the cutoff point.
The epiderm, endoderm, mesoderm, and ectoderm are. So, yeah, I tried to stay out of the religious portions of it. I tried to stay just in the science. And, you know, I know that when we talked about the primitive streak, I know that I saw that of the major religions like Judaism, Christianity, Islam, and Hinduism, there was a rationale put forth by all four of those religions as to why that time period of the primitive streak was acceptable. But I think that many religions concur that primitive streak, that there can't be anything done beyond that, or that's human experimentation.
Okay.
But you know, I don't want to in any way portray that I have any religious knowledge of this. I don't. I just know the scientific end of it.
Okay. Simon and Manuel, any questions?
Yes, thank you. If I might go first. First, an update on the case last week I talked about with Dr. Obokata, who had done her PhD thesis on the stem cells that you could, stab cells that she said if you applied heat or stress to cells that they would revert back to stem cells, and this was published in Nature, and it was retracted, and she actually Waseda University took away her PhD this week. There's a big news in Japan.
Yeah.
Wow, that's a big news, Simon.
That's big news.
So you sound like like a reporter.
I know.
Well,
I'm actually going to.
This is news. You're being active out in the field.
I'll put on my English show.
Didn't someone commit suicide?
Yes, her boss.
Yeah,
in Riken.
Her boss.
For Riken Institute to take responsibility.
Because research was considered to be flawed.
Right. But my question is regarding, well, this is regarding the limits of medicine, really, and concerning your stem cell presentation. So this morning I was sitting next to his radiologist, and we were looking at different cases, and we had a case this morning which was an acute infarction of the medial pons. And we were looking at flare images and diffusion weight images, etc., and then a micro angiogram. And we came to find out that it's probably due to a problem with a paramedian artery. And I said, what can we do for this person? She said, probably nothing. And she said, what do you suggest, Simon? I said, well, at some point could stem cells, iPS cells be used? She goes, well, how are you going to get in there? And I was stumped. I didn't know.
Well, remember, we talked about that because the idea is, what route are you going to deliver the drug by, or or the cells by? Like, how are you going to integrate the cells into the substrate? Like, how are you going to get the cells to function in the arterial system of the pons? Because the pons has these. It has short feeders and really long feeders, and a stroke in the center of the pons is right there in the middle of everything. So even if you were to produce a cell that was exactly like the cells in a basilar artery aneurysm, like, how would you get it there? And
right.
And one cardiologist who was working, one researcher from Carnegie Mellon that was working with a friend of mine at the University of Pittsburgh years ago, his answer was that they wanted to put these genes and these substrates on cardiac stents. And
Wow.
I would have never thought about that. And that's what they did. That's how they got the idea for drug-eluting stents and the idea for trying to get a substrate into the pons. Probably the only way to do it is going to be endovascularly, right? Or, how else are you going to deliver it?
Let's see.
So that it's not only did you refine the cell right so that all the HLA types are matched, but how are you going to A, package it, and how are you going to B, deliver it, and how are you going to know that it's going to integrate? It's like I was watching this YouTube video, and I had seen it many months ago, but it just appeared again somewhere else. And there's a neurosurgeon from Italy. I think he's a neurosurgeon who is talking about doing a head transplant on a human. And first of all, the patient in question is somebody who has Wertig-Hoffman's disease. Now, Wertig-Hoffman's disease, as far as I knew, it's an awful disease, and I've never heard of anybody living much past 10 months with Wertig-Hoffman's disease. So he's claiming that the person whose head he's going to transplant has Wertig-Hoffman's disease, and the guy's like 30 years old, and he's mapping this surgery out, and he thinks he's going to do it in 2 or 3 years. So I must say that sounds a little specious to me.
And spinal cord.
Yeah, and I don't know. I said to my friend, I said, how is he going to, I said, things are probably going to go south in a hurry when he tries to anastomose the brainstem to the spinal cord. Things are going to go south in a hurry. Wow. My friend said, well, maybe he'll just use glue.
Glue, yeah.
And the person seems so ebullient that I have to believe this is a joke, Yeah.
Right.
National Enquirer of Neurosurgery. Manuel, do you have any questions or comments?
Yeah, this is a very interesting topic, also somewhat controversial. My questions are from the practical point of view and focused on neurodegenerative diseases such as Parkinson's disease, where some trial where some experiment has already been done. But I have some questions, some doubts, because here in Parkinson's disease, for instance, you know how to put stem cells where you're going to place them, because you have deep brain stimulation and you have the pathway, you know the pathway, how to put, you know the methodology to put cells in the substantia nigra or in the basal ganglia. But my question is, how do you make stem cells differentiate into dopaminergic cells and then how do you integrate those cells in the circuits? Because one thing is putting cells in the correct place, which I think it's possible today, but how do you make those cells functionally, from the functional point of view, integrate in the circuits? And moreover, will that make a difference? Because the disease still continues.
No,
The neurodegenerative process still continues. The tau protein, alpha-synuclein protein, will keep damaging cells. So will that make a difference?
well, that's an excellent point, and I want to say right off, up front,
Okay.
I'm a moron compared to like Andres Lozano in Toronto. He's the guy that's mapping out. You know, he's done so much research on the physiology of Parkinson's disease because, you know, and we did talk about this last week. And what you said is an excellent point. You know, we know where the substantia nigra is, but all the physiology of the Parkinson's disease, and that's something that Andres Lozano is mapping out now. You know, it is what happens in the whole physiology of the balance of the dopamine, and you know those cells. Most of the time, people get symptomatic with Parkinsonism, and 80% of those neurons are already gone. So they've already had the disease a long time.
Right.
But the most interesting thing I saw about stem cell research as addressing the target of Parkinsonism was that it's very unlikely that anybody is going to approve a human trial for treatment of Parkinsonism with stem cells anytime in the near future, because it's the results that we're getting from deep brain stimulation are unlikely to be surpassed by stem cell implantation. Because remember in the late 80s or early 90s when there were several people in Mexico doing quote-unquote transplants and implanting dopaminergic tissue into the brains of some people, and I don't know how, I can't imagine that was really an approved trial. But you know, that's the point, is if you take a dopaminergic cell, where are you going to put it? Are you going to put it in the striatum? How would you possibly put it in the substantia nigra? I mean, how do you incorporate that cell so it functions? So I understand what you're saying. I can't answer any of those questions, but I have the same questions. But it does sound like, you know, the stimulation of the subthalamic nucleus yields very good results in terms of the movement disorder. But you know, for somebody with a Parkinsonian plus syndrome, or who has more of you know, bradykiniasias, I don't think they're going to be offered all that much from deep brain stimulation. But I think it's probably true that the stem cell, the idea of using stem cells for Parkinsonism right now, we're kind of far away from that, I think.
Yeah. You know, Richard, let me just comment. We've done a lot of hangouts with the Parkinsonism, and Manny knows him too, Dr. Abdul Rana from Toronto. And maybe he knows that neurosurgeon. What's that neurosurgeon's name again?
Andres Lozano's world-famous neurosurgeon whose chief interest is Parkinsonism.
I'm sure they know each other.
There's nobody that doesn't know Lozano.
Lozano, okay.
Yeah, and I mean, he's like, you know, Irving Cooper was the go-to person in the 50s and 60s for stereotactic movement disorder surgery and Parkinsonism. Lozano's one of the big thought leaders in, you know, neurosurgery for Parkinsonism.
Okay, well, try to reach out to him and get him on a
Oh,
hangout.
he's published so much, but I mean, he's not doing, he's not doing stem cell therapy on humans or anything like that. I think the results he's getting now are pretty decent, and it's like people said, it's unlikely that any result we get with stem cells is going to surpass what's already a pretty safe technique by stimulating. So I agree with everything you said, and we did talk a little bit about it last week. I have no idea, just recreating a neuron doesn't mean there's any way to hook that neuron up and make it work.
Right.
That's a big deal too. Many years ago, there was a drug company in Ireland, and I thought they were going to take over the world. Their premise wasn't on inventing new drugs; it was on a new way to deliver drugs. And
Right.
the delivery mechanism is just as important as the substrate you're trying to deliver. Because if it doesn't work, you might as well be waving a chicken wing over somebody.
Yeah. Well, let me just remark that I heard from a researcher that doing stem cell research is a lot easier in Canada than it is in the United States. It's less restrictive. Have you heard anything about that, or?
Well, according to what I read, there is not a single law against any stem cell research in the United States.
United States, okay.
It's only there are restrictions on how to, how you can attain grants for the research.
Right.
You can't. It's public money, especially NIH money, which is citizens' money. So.
That's a big source of funds for a lot of
gigantic source, but apparently if somebody has the money or the backing, they can do pretty much any research they want on stem cells. There's nothing restricted. It's the financing of the research that's restricted.
Mm-hmm.
So, but I also don't know anything about the Canadian research now. So who knows? Maybe I'm wrong. Maybe they encourage something we don't. I don't know about that. I'm just doing a book report.
Okay. Okay. Well, it was a good book report. Okay. I guess we'll wrap it up. That's part 3. Are we going to do part 4, or that ends the basic series?
I think we tackled enough about
most of the basics. Okay.
Yeah, but I think we ought to try to use some of the basics, and I'm sure the opportunity is going to grant itself, because there are all kinds of products being introduced as amniotic fluid that contains stem cells, this, that, and the other thing. And, I remember years ago when something was marketed to prevent scarring.
Mm-hmm.
And my favorite article about it was entitled Amniotic Fluid. It's not just fetal urine anymore. Because there's no... Science was nonsense.
Well, some people consider placentas a delicacy.
Yeah,
Oh man,
I want to stay out of any country.
Yeah, we'll leave it at that. Okay,
Yeah.
Richard, thanks a lot for another excellent presentation, and thanks to Manny and Simon. So we'll just end this, and thank you very much.